From Imperial College London, welcome to this special lecture vodcast.
Well, Richard, thank you very much. It's a tremendous honour to be here. I hope to keep you entertained for a little while. I'm going to start off simply and proceed until it gets beyond me. I did come here once to meet Sir Ernst Train. I think it was 1973, about the time he retired, and he'd read something about these endogenous retroviruses, viral genomes that are inherited as Mendelian traits in the host, and wanted to know more about them. So I was sort of summoned to his august presence in the fermentation laboratory, I think it was called, and he quizzed me, and so that was a great meeting. But in the past 8 years that I've been back at UCL, it's been an enormous pleasure that Benny Train, one of his sons, is one of my colleagues, and it's great to see Benny and Julia Train here tonight. And Benny sort of occasionally says, "Robin, you don't really understand immunology," which is quite true, and sort of helps to keep the sort of intellectual sort of a level of our whole department. So that's another added pleasure. Of course, there's lots of friends here, lots of friends at Imperial, including 3 members of the faculty who once at some time in their earlier careers had worked in my lab. So I feel I've given an Imperial something, Richard. Now, this prize lecture should really go to a musician, I feel, because that was Ernst Train's passion and still is Ralph Cohn's passion. I love classical music, but I sing dreadfully out of tune. And I would not impose a CD if I ever made one, or anything else. But let's see if this works. I thought I'd give the lecture in sonata form, yes, especially for Ralph. So there's a slow introduction, of course, and then we'll speed up a little. And the first subject or theme will be on the receptors for HIV. And then the second subject on the neutralizing antibodies, because it's really looking at the same thing of how HIV interacts with cells. And then we'll really mix them up in the development. I won't give a recapitulation, just a couple of conclusions. But a coda at the end, just a twist of using the same theme to study other viruses. So that's the structure of the lecture. So what about the introduction? Well, just to remind you that AIDS is a new disease, that it was first recognized only in 1981, that Françoise Barré-Sinoussi and Luc Montagnier discovered the virus just 2 years later. And I came into the field, I remember the exact day because it was leap year day. It was February the 29th, 1984. And a postdoctoral scientist in my lab, Rachini Cheongsong, who Jonathan will remember well because she worked here at Imperial for several years later, came back from France with a sample of the virus provided by Montagnier, and we began to grow it. They'd had trouble propagating the virus in France, and I said, let's try some cell lines, T-cell lines. And it took off like a rocket. So we gave it back to them a month later, and they said, wow, this cell line's producing, 100 times as much virus as we could get before. And that provided the early antigen for screening tests, which were based on antibody tests. And we had an exciting time in which Jonathan was young, but one of the leaders in really looking at which risk groups were infected in the UK. And it was around this time that there was a suspicion that there was something like AIDS in Africa. No one was quite sure. And we worked with David Sawada, Nelson Siwukambo, and Makerere Medical College in Kampala in Uganda, and they sent us samples. And sure enough, the patients with so-called slim disease were seropositive. But we thought the test wasn't as specific as it looked to be with British sera, because about 10% of the controls, who were hospital staff, nurses and doctors, were also giving positive results. And I remember the evening when it really sank into us that the test was specific and that there was a 10% rate of infection. And that was one of the most sort of galling moments in my career. And sadly, although Uganda has some control over the rates of infection through public health policies, of course we now know that in some other African countries the rate of infection is much higher. However, I put this list up because I don't think it's all bad news about HIV. I think the scientific study of HIV as a virus has paid off in some public health dividends. As I said, just being able to grow the virus before we had recombinant antigens gave us blood tests. And within 2 years of having that first rather tentative antigen test and antibody test, all the blood banks in industrialized countries had introduced screening of all blood donations. That was pretty fast development. The first antiretroviral drugs, idovudine, had been noted to inhibit murine leukemia virus some years before and was taken off the shelf and gave wonderful results for about 3 months. And then the virus developed resistance in the first patients. But it showed that you could knock the virus back by understanding its replication. But it took another 10 years before the combination of antiretroviral drugs really made a big difference. We've had big advances in preventing mother-to-child transmission. Microbicides, vaginal microbicides, there's a lot of effort, including here at Imperial College. That's been more problematic. And the big failure, if you like, in the whole of the scientific community has been developing a vaccine. And I don't think it's because we're dumb or because not enough money's been thrown at it. It's just difficult, just as making a good vaccine to malaria is difficult. But we must go on trying. So if we look at a very simplified life cycle of the virus, here's a virus particle, HIV. Here's a white blood cell, a CD4 positive lymphocyte. And like all viruses, it's got to get inside the cell in order to replicate. They're intracellular parasites. So it's got to dock onto the surface that I'm going to talk about, get inside. The reverse transcriptase of its RNA turns into DNA. The DNA integrates into chromosomal DNA. And then the transcripts of RNA can be translated as messenger RNA into proteins or full-length transcripts packaged into the next generation of virus particles. And again, understanding this retroviral life cycle has given us points of attack for therapeutics. The first drugs were blocking reverse transcription, then the protease inhibitors blocking maturation of the particles emerging from the cell, and now we have fusion inhibitors and integration inhibitors as well. This scientific knowledge has translated into practical means. And to illustrate that, let me show you some data that I've taken from the CDC in USA and adapted. And this shows the mortality rate in the US from AIDS, sort of coming from nowhere, first being seen in a handful of patients in 1981. And within 10 years, it had become the leading cause of death in an age group that doesn't expect to die, youngish adults. And you can see the other things, 4 leading causes of death. And then it's plummeted down here. Now, that's not because people stopped catching HIV. It's because the combination therapy of antiretroviral drugs began to be applied then. And it led to about a 75% drop in mortality rates and in morbidity, too. The wards that were full of patients with AIDS emptied, and most treatment is now given in outpatient clinics. So it's made a tremendous impact. Now, unfortunately, we can't deliver this all over the world, not yet. So if we put the same US curve on a world scale of incidence of AIDS in millions, we can see what's been happening in Africa and now in Asia, and there is no real impact of antiretroviral therapy. So there's a big challenge to us that's not a scientific challenge, not something for a virologist like me to tackle. How to deliver this therapy and the complications of treatment to the rest of the world. Now, to theme 1, the docking of HIV. Well, let's imagine that HIV is a bit like a space capsule trying to dock onto a planet. So it's got to get inside cells to replicate. It's in the bloodstream. How's it going to get there? So it's going to zoom up to a planet, and what does it look like when it gets close? Where's it going to land? Well, in Africa, of course. Now, this is really just to remind you that the cell surface here is very complicated. It's bristling with different molecules that are sticking out from the cell membrane, different antigens, glycolipids, particularly glycoproteins, and other structures. Viruses have to dock onto something and use something to get into cells. Now, whatever that something is, isn't there just for the convenience of viruses. Viruses as parasites are going to hijack something that has some other function to get in. So which one is going to be the docking receptor for HIV? Well, it's the CD4 molecule, and I've drawn it as a hippo here. One or two of you have seen this once too many times, I'm afraid, but here it is. And we found this by working with Mel Greaves at the Institute of Cancer Research, where I was at the time. It's great to see Peter Rigby here, and Peter Beverley. And Peter Beverley had the whole world's collection of leukocyte surface antigens. He'd just come back from the international workshop, and we tested them blindly. We went through about 155 of these, and we picked up 14 that blocked HIV getting into cells. And when we decoded them, and this was work of Gus Dalgleish and Paul Clapham, we found that all 14 of them were antibodies to CD4, whereas the others didn't block. And this immediately explained something about the disease, because we already knew from the the first full descriptions of AIDS as an immune deficiency, that it was a specific deficiency of T helper cells. And immunologists identify T helper cells as being CD4 positive cells. It's very curious that the virus happened to use the same marker as the immunologists used, because they could have used a dozen other markers, but the virus must have been reading the textbooks. It wasn't called CD4, it was called T4 in those days. And it also helped to explain an important paper from David Klutzman and Montagnier that came out a couple of months before ours, that they took these lymphocytes in culture and sorted into populations that were enriched for CD4 or enriched for CD8, and found that the virus was growing in the CD4 cells. And here we showed, well, it was growing in those because those are the ones it could get into by landing on CD4. Now, today we know about lots of cell surface molecules that are receptors, but in fact that discovery of CD4 was the first protein receptor for a human virus to be discerned. So it sort of set off this kind of trend of looking for receptors. And one could begin to map where on the CD4 protein the virus bound. First, we could map, I'm not using a pointer, the antibodies that seem to bind to the N-terminal domain of CD4 were the strongest blockers. And the two that didn't bound to the domain that was most proximal, still outside the cell, but proximal to the membrane. One could also look later on when the gene, or the cDNA for CD4 was cloned a year later in Richard Axel's lab by Dan Littman and Paul Madden. We worked with them to look at and express clones of the receptor. And a single mutation in the N-terminal domain of transforming a phenylalanine to a valine at position 43, which gave a sort of floppy-eared hippo. It just changed this epitope, single amino acid. There was absolutely no binding and no entry. And in fact, before this, Myra McClure, who's here tonight, did we did what I thought was a smart way of site-specific mutagenesis. We looked at CD4 lymphocytes from about a dozen different species of primate from the zoo, and looked at natural evolution and which ones could bind and which ones couldn't, which was sort of nature's way of looking at site-specific mutagenesis. But this was smarter because it gave us a firm answer. So there was quite a lot one could do in those ways. Now, by this time you must be thinking, have you really come to a scientific lecture or a cartoon show of hippos? So I want to show you how immunologists depict CD4. And they depict it as a rod with 4 immunoglobulin-like domains. And if you take off the top one, that's what I'm calling a hippo head. However, it was later crystallized. And if you look at a space-filling model of the crystal and rotate it in the appropriate way, I think it looks more like the head of a hippo than it looks like this. And this is the phenylalanine out here. Okay. Now, one can take those soluble domains, express them not hooked up to receptor, and if you saturate HIV, it works as a wonderful inactivating agent. It neutralizes HIV. So there was a lot of excitement in the late 1980s that this could be developed as a drug. But we found that although this soluble CD4 potently neutralizes lab-adapted strains of HIV, David Ho then showed that it was only very weakly neutralizing for primary strains. However, a different kind of structure, CD4 hippo head, or actually the top 2 domains of CD4, linked up to the Fc portion of immunoglobulin, is very potent at neutralizing HIV, even primary strains. But this is a very big protein molecule to consider using as a drug. It's not really on, although it does the job. Another thing we found was that while CD4 was necessary for HIV to infect cells, it was not sufficient. And this was work with Richard Axel's lab, where we took the gene for CD4 and we transfected it into human cells, such as HeLa cells that wouldn't normally express it because they're not cells of the immune system, and also into mouse cells. We found if we looked at binding of the outer envelope glycoprotein of HIV by ELISA, it bound very well, both to HeLa cells and mouse cells. But if we looked whether you obtain cell-to-cell fusion or penetration of pseudotypes, I'll come back to those, or HIV replication itself, it worked for human cells and not for mouse cells. And if we looked at heterokaryons, cells, mouse cells expressing human CD4 but fused to HeLa cells, then they were susceptible. So there wasn't a dominant inhibitor in mouse cells. There was something lacking in mouse cells. We published this in 1986, and a lot of labs started looking what was the lacking receptor? Was there a co-receptor? What could it be? And it took 10 years before they were found. And what was it? Here's the beast, a 7-transmembrane G-protein coupled receptor. The first hint came from Bob Gallo's lab, where Fiorenzo Cocchi observed that certain chemokines, these three, would block HIV infection. Didn't quite understand why or how. And very soon afterwards, Ed Berger at NIH used expression cloning techniques to show first that lab strains would dock onto this chemokine receptor, CXCR4, and primary strains of HIV would dock onto, interact with CCR5. So the virus has to bind to the hippo head and then somehow interact with this receptor in order to get inside. And as I said, these receptors are not sitting on cells merely for the convenience of HIV to come along, but they have natural ligands. CD4 interacts with the major histocompatibility class 2 antigens, and here's where the link between those discoveries comes, that these chemokines bind to CCR5, whereas a different one, stromal-derived factor 1, binds to CXCR4. And all the primate immunodeficiency viruses recognize CD4. So does human herpes virus 8, oddly enough. And we now call the primary isolates that interact with this receptor R5 and with this one X4. That's the jargon in the HIV field. What actually happens on binding to CD4 and then interacting with CCR5 is very complex, because the HIV GP120 undergoes an induced fit with CD4. CD4 kind of recognizes it, and it then folds around, and CD4 sort of plugs into a cleft in the glycoprotein. And that then opens up the site on the same glycoprotein of the virus that interacts with the co-receptor CCR5. And so in these models from Steve Harrison, see the CD4 binding site is the green one. After binding, it sort of formed a tighter circle, but it's down in the cleft that the CD4 really goes. And that brings two different parts of the molecule here together to make a contiguous epitope, which is the CCR5 binding site. Now, some of you will have heard John Scales' chain lecture a couple of years ago, where he showed much more exquisitely how these conformational changes occur with the influenza hemagglutinin. And I must say, the HIV people always following the example of influenza. The influenza glycoprotein has been the model for all envelope viruses all the way. So if we summarize this and look at the virus coming down onto a cell membrane, the glycoproteins or spikes on the outside of the virus are in a trimer. There are two proteins in each bit of the trimer, a GP41, which is the tethering protein to the lipid envelope of the virus, and GP120 out here. And the virus keeps its receptor binding parts rather tightly closed. On interacting with CD4, they open out, including the pink bit which interacts with the co-receptor, and this allows the GP41 to come through and a hydrophobic domain to form here that plugs into the host cell membrane. This then folds back into a 6-helix bundle, draws the two membranes together that fuse and allow the contents of the virus, which is up here, to pop into the cell. So it's a lot of different molecular interactions going on with rather complicated energetics. And it's also given opportunities for drug development beyond CD4, more practical drugs, of which one has been licensed in the clinic for 3 or 4 years now, which is the T20, a synthetic peptide which is homologous to these green bundles and binds onto them and blocks them going further. It really binds here but blocks this last step. And there are a number of other ones that have been in development. And I believe at the recent CROI meeting 2 years ago, one of these got the thumbs up. Perhaps Janet could tell me afterwards, because I wasn't there. She's shaking her head. But the rumor was that it would get the thumbs up. One lives in hope. So that's the docking and entry of HIV. What about my second subject? Well, that's HIV neutralization. Let's see if we can block the docking with antibodies that neutralize HIV. So here's some HIV particles, electron micrographs. These are the trimeric spikes, and these are just diagrams of antibodies blocking it. And the dogma, according to Dennis Burton and Paul Parran, who are the leaders in HIV neutralization, is that anything that binds to native virus, if it saturates these glycoproteins, it neutralizes. That binding is neutralization. Now, there are a lot of antibodies that bind and don't neutralize, but they're thought to bind to denatured spikes. I don't quite swallow the whole story, the whole dogma, but certainly those that do neutralize bind tightly, and you need to saturate the surface to get complete neutralization. When we first started working on HIV, we looked at neutralization. We had been working on the other human retrovirus, human T-cell leukemia virus type 1. And this is from our first paper on neutralization. And if we looked at antibodies to envelope by ELISA to the envelope glycoproteins, or by membrane immunofluorescence, or by neutralization, in the case of HGLV1 infected patients, we got high titers in all cases. If we looked at HIV1, we found similar titers to envelope glycoproteins by ELISA or by immunofluorescence, but very low titers by neutralization. So here was a warning right from the start that attempts to develop vaccines that gave humoral immunity was going to be a problem. And we also found a year later that antisera raised against recombinant GP120 gave nice neutralization against the strain from which the recombinant protein was derived and didn't touch any other strain at all. So we knew that experimental sera, vaccine sera if you like, wouldn't cover all the HIV strains that were out there. And we haven't moved very far in the last 20 years to improve this situation. If we look at the structure, there are different models, but here's the first decent structure that was obtained for GP120. It's a very difficult protein to crystallize because it's got these floppy loops moving about. But Peter Kwong's group at NIH have been one of the pioneers in this. And by truncating them and then just sort of modeling them in, they got a core structure here. This is the monomer of GP120 with its outer face here, interacting with its three partners in this inner face. And there's a couple of things to point out. It's a very highly glycosylated protein. These light blue and dark blue structures are sugars that are attached, N-linked and O-linked sugars on the glycoprotein. And they almost form a protective carapace. They sort of ward off the antibodies and cover up the receptor interaction sites, as do these loops. If we look at a more recent simplified model of the trimeric spike here, here's GP41, the three strands, and here's GP120. There's a CD4 binding site, the V3 loop, which is subject a target for neutralization, but strain-specific neutralization. After CD4 binds, then it opens up, interacts with CCR5, and then you get the fusion. And for all the effort of studying neutralizing antibodies, and there are probably 50 or 100 different antibodies around, there are only 5 that cross-neutralize different strains in different subtypes or groups of HIV, HIV-1. And all of them were discovered some 10 years ago or more. So that there's very few that can see common sites that are common to all the viruses. One of the problems is that HIV is variable. We all know there's lots of strains out there. But it is highly variable. So we think of influenza as variable. And each year the WHO has to pick the strain it thinks is going to be epidemic in the coming winter and hope it's got it right, that there hasn't been too much antigenic drift, and prepare a vaccine. Well, this is some data derived from Betty Corber at Los Alamos, where the length of each spoke or spike is an indication of variability of either the influenza hemagglutinin or the GP120 of HIV. And we can see the length of the spokes in a single individual who's been infected with HIV for 6 years is about the same as the global variation of a strain of influenza, of a current strain of influenza. Now, here's a virus that has been one serotype for the last 100 years, measles, and we got a vaccine that works very well, thank you. But here's the problem. If we look at the Congo, for instance, which is close to where HIV first started, has had longest to keep going, we have all these different clades or subtypes, and each one is variable. But each one of these viruses has to dock onto CD4 to get inside the cell. So a vulnerable point that should be constrained in its variation is the CD4 binding site. And there's a beautiful paper that appeared just last month, February the 15th, again from Peter Kwong's lab, where he's got the first crystal structure of the core of GP120 with the best cross-neutralizing antibody, B12 of Dennis Burton's, binding to the CD4 binding site. And here's CD4 bound itself. So the B12 is covering part of the CD4 binding site. CD4 is more extensive, but this is the cleft that it gets into. And we thought if we can develop antibodies that really got into the center of the binding site here, we might get more broadly neutralizing antibodies. And we've begun to go about that. Now, how are we doing that? Well, we're using llamas. The Gates Foundation, its publicity, said we were infecting llamas with HIV, which is a little bit beyond what we were doing. So I got a lot of emails from animal rights people saying, how dare you? And we're immunizing llamas with inactive GP120, GP140 proteins, which is no worse for the llama than its normal immunizations against brucellosis or whatever. This is work in collaboration with a biotech company in Belgium and colleagues at the University of Utrecht. What have we done? Why llamas? They look sweet, but in fact they spit and bite and kick. Well, llamas are a curiosity of mammalian immunology. They make conventional immunoglobulins, an IgG like this that has 2 heavy chains and 2 light chains. But they also make heavy chain only antibodies, which function as antibodies. They have all their antigen recognition in this N-terminal part of the heavy chain. And this was first shown by a Dutch group more than 10 years ago. This means you can make phage libraries, and you don't need Greg Winter's technology to make single-chain antibodies, because the llama's done it for you. And you can clone off this top end. And another advantage of it is it has a long CDR3 loop. This is an important part of the antibody for binding to antigen, and particularly finding antigens in clefts. It reaches further than other antibodies reach. And it's small. You can express these as 12 to 15 kilodalton proteins, and they're stable. You can make them in gram amounts in yeast, and they still work as antibodies. So we were funded to do this in the hope that they might be potential microbicides. Well, we don't know that yet. I'm a bit suspicious of any protein being a microbicide. But they're not very immunogenic, and you can select them. Having selected your antigen recognition, you can then select them further in phage to be stable at both pH 5, which is about the pH of the vagina most of the time, and pH 7.5 to 8, which is the pH after ejaculation, because semen is very alkaline. And you want an antibody that's going to work in both situations. So they might be useful. At the least, they're going to provide 2 doctoral theses for my students. So to get llama antibodies that are going to hit the CD4 binding site, it's very simple-minded. It turned out to be very tricky. You come in with your phage library, very complex phage library, and there are going to be lots of antibodies that bind all over GP120. And you want to select those that see the CD4 binding site, which I've made into this cleft here. So you come in with soluble CD4 in excess, and it's going to dislodge just those phages that it's competing with. And because the phage contains the gene that's coding for this little llama antibody fragment, you can then selectively grow it up. You go through about 6 cycles of this and lots of washes, and the students stay up all night. And eventually you get something that works. Now, what we got so far was from llamas that were immunized with the clade C virus. It turned out not to be a particularly good recombinant protein, but it was what was available, CN54, made by Simon Jeffs at Imperial, I believe. And thank you, Simon, if you're here. And he's making more for us now, and he's getting funding through our Gates project, so that's bringing something into Imperial. And then we selected on 3B, which is a clade B virus, and then got some that cross-neutralized, some clade C and some clade B. Now, actually, I was pretty disappointed when we were analyzing these data, because I wanted ones that cross-neutralized a lot. But when we went to the annual meeting of Gates, the first time all the vaccine-supported people were there, Bart Haynes, who runs CHAVI, which is an enormous sort of consortium of grants on neutralization, was showing his data, and I realized that he was using an inhibitory concentration 50. He counted a positive, a hit, as anything that neutralized 50% of HIV. And we were using 90%. If we look at 50%, there isn't a single virus that gets away with it. So he's an optimist, and I was a pessimist. Now we're sort of somewhere in the middle. Well, let's go on to development. Now, Ralph, this is a Fugato section. I've not talked about this before, and I'm not very good technically. So if the audience gets lost, it's my fault, not you. What about genetic susceptibility to HIV and AIDS? All humans vary. Some are going to be more susceptible than others. We know that they're so-called good and bad class 1 and class 2 MHC antigens, and Andrew McMichael touched on that during his train lecture. But I want to talk about the genetics of the receptors. CD4, there is one polymorphism. It's in the 4th domain near the membrane, and it doesn't affect CD4 binding or function. In the same year as CCR5 was discovered to be the co-receptor, some people were found who were homozygotes for a deletion of 32 bases, which put the whole thing out of frame and made really a nonsense truncated protein. And these were found because people were puzzling over couples where one was infected, the other person in the partnership was not, and they should have got infected because they'd been having unprotected sex, sometimes for years. And some of those turned out to be the ones who didn't get infected, were spared the infection despite exposure, to be these homozygotes. About 1% of the Caucasian population are homozygotes. This mutation does not occur in Africans, and the further north you go in Europe, the commoner it is. In Scandinavia, some 18% are heterozygotes. Now, if you're heterozygous, you have one wild-type CCR5 and one mutant, you express less CCR5 on the lymphocyte surface, and if you're infected, it takes longer to progress to AIDS. It's sort of partly protective or delays progression to disease. There are also mutations in the promoter, not the coding region of CCR5. And these are interesting in conjunction with not mutations, but gene copy numbers of the major chemokine CCL3L1. They've changed the terminology of the chemokine people, so I have to get used to this, which binds to CCR5. And it's the combinations of these that really paint a real picture in progression to AIDS and in susceptible infection. And I'm going to come on to talk about Duffy. So to summarize an extraordinarily dense and complicated paper that I asked students at UCL who were doing the advanced genetics course to dissect, and they gave up. This is Sunil Ahuja's group, who we're now working with. And he's brilliant, but it takes some following. The gist of it, if I've interpreted his work correctly, is that if you have a low expression of the receptor on the lymphocyte surface. You only have a few of these molecules around, and you have a large amount of plasma chemokines, which is what Fiorenzo-Cocchi first observed. Then together, few receptors, lots of competing chemokines, then there's slow progression to AIDS. On the other hand, if you've got lots of receptors, the promoter mutations allows higher expression, but rather few copies, duplications of this gene, so you have less chemokine. Then there's less competition with HIV, and you get rapid progression. And it may be that HIV's got this high affinity docking to CD4 to help it get there and outcompete the natural ligand too. Because we suspect that that's a rather, recognizing CD4 is a later evolutionary step in lentivirus development than the chemokine receptors, without too much hard evidence. Now, what about Duffy antigen? Some of you will know about Duffy as a peculiar antigen on red cells that's got something to do with malaria, and that's about all I knew about it, until I realized it was a chemokine receptor. Now, AIDS researchers since 1996 have studied every chemokine receptor to get hold of, with the exception of Duffy. And that's odd, because there is a major polymorphism that distinguishes Africans from everyone else in the world, and that is expression of Duffy on red cells. It's normally expressed on red cells and endothelial lining cells of blood vessels. It's regarded as a sink for a number of chemokines, CC and CXC chemokines, but we're not sure it is. It's known to be the receptor for Vivax malaria, for the merozoites that cycle in the red cells. And as I say, most Africans lack dark on the red cells. So it's another 7-transmembrane receptor. Here's a fax plot of my red cells. And the further that this red peak shifts to the right, the more Duffy there is on red cells. And this is a fax plot of a Ghanaian friend of mine who's Duffy negative, using a monoclonal antibody to the Duffy or dark antigen. Now, there was a paper published in The Lancet 5 years ago that noted that HIV is associated with red cells. You can take red cells out of an infected patient, you can wash them, and they've got HIV on them. HIV RNA can even be detected on red cells from infected people when they have very low free plasma loads of virus. And it tends to be associated with a bad outcome. So I asked myself, what's it binding to? Could it be Duffy? And we found, this is work of Stuart Neal and more recently Ed Wright, that HIV-1 indeed binds to red cells, and it binds to dark Duffy on red cells, but it can't use as an entry receptor. With HIV-2, we tested a dozen strains, isolates, and it also binds to red cells. But when we transfected the dark gene to indicator cells that can be infected with HIV if there's also CD4 there, we found that about half those strains could also use Duffy as the entry receptor, not just a binding interaction, but a fusion. Well, so what? Well, there's been a lot of discussion about HIV binding to DC sign on dendritic cells, and binding to red cells may be doing something different. They go into the spleen, they jostle up against lymphocytes, and it may be helping them, helping to deliver HIV into cells. So we thought we'd better look at Duffy negative red cells. Here's 5 strains of virus, and we've standardized the binding to my red cells, and my Duffy negative friend has much less binding. So Duffy is playing the major role in the binding. If we look at inhibition, here's a chemokine that binds to Duffy, so it can outcompete the binding of virus, which we measure then by the delivery by adding the red cells to lymphocytes and seeing how much infection you get. This is an isoform of MYC1 alpha that does not bind to Duffy. If we use antibody to Duffy, we can block it. If we use neutralizing antibody to the virus, we can block it. And here's a genetically Duffy negative red cell again. Now, what is the genetics behind this phenotype? There's a mutation at -46 before the start site of coding. And this sits in a GATA1 box that this transcription factor, GATA1, has to bind to this to allow expression in red cells. It's a very specific red cell transcription factor. People who are homozygous for this mutation, who have TCs instead of TT or TC, then don't make it on Duffy. There's also a polymorphism here, which is in the first extracellular domain of the 7-transmembrane receptor. And in fact, we think this also affects HIV infection, but we haven't really worked it out yet. So we started working with Sunil Ahuja, the geneticist in Texas. And first we looked at the distribution of these genotypes in a DNA collection from Ceph in France. And you can see here, these are Africans. You can see that very few Africans are not homozygous for the mutation that doesn't allow expression on red cells. On the other hand, East Asians, Europeans, Americans, Native Americans, they all do. In the Middle East, there's a bit more Africa in the Middle East than some of the Middle East people would like to think. Just as with African Americans, only 70% of black Americans are Duffy negative. So there've been more white genes getting into the droit de seigneur of slave owners and so on over the years than their social identity might suggest. So let's ignore the coding mutation and just look at these. We looked at a large cohort of Americans that include a large number of African Americans, a large number of HIV positive, that has been established by Matt Dolan at this medical center for American servicemen. And this was the cohort that Sunil Ahuja had already been studying for the CCR5. And so we collaborated on dark polymorphism. And we get some surprising results that we don't fully understand. First of all, Duffy negative people, that's most black Africans, are much more susceptible to infection with HIV than Duffy positive people. Now, this 150% rate, no, it's or you could put it the other way around, say Duffy positive people are a third less likely to get infected. That's a similar risk factor to, say, protection from being circumcised. And no one's observed it before. The paradox is that if you do get infected, then Duffy negative people have a slower rate of progression to AIDS. It's counterintuitive. And I'll show that because the statistics are robust. There's a Kaplan-Meier plot. So here is the curve over the first 6 years of Duffy negative black Americans, essentially, who are homozygous for this mutation in the GATA box. And here's the plot for those who are either homozygous wild type or heterozygous. This is CD4 count rather than AIDS. And this effect is larger than being heterozygous for delta 32, the Caucasian mutation. So it really is a remarkable finding. We're still puzzling over it. What does it mean? Well, what could it mean? Ed Wright and I were trying to work this out. Well, risk of infection, we do know that high levels of chemokines that bind to CCR5 tend to be protective against infection. You're less likely to get infected. The dogma is that Duffy is a sink that removes chemokines, so you'd expect lower levels if you're Duffy positive. But we think, from one little report on a knockout mouse, that the opposite is true. That there's quite a good on-off rate between chemokine and Duffy receptor, and that if you have these Duffy antigens, it's keeping the chemokine in the peripheral blood and may elevate it. And we're setting up to study this, if we can, in blood donors. The ethical permission, even though it can be totally anonymized, but we do need lots of blacks in there, is a bit tricky. And just doing a back-of-the-envelope calculation, only a very small minority of T helper cells in the blood circulation are CCR5 positive. There's many more in the gut-associated lymphoid tissue, but in the blood there's few. And in any case, there's about 100 times as many red blood cells as there are T lymphocytes. So there's probably an excess of 10 to the 6 Duffy for every CCR5 receptor around in the peripheral blood. So there's some serious competition there. But again, CD4 probably plays it well. This can grab hold of HIV and bring it close. And another view that Sunil favors is a hypothesis on progression, that you have infection, that actually free virus in the plasma is very unstable. We don't know really what the half-life of an infectious particle is. We know the kinetics, dynamics of production and turnover, but we don't really know how long a virus can last. But it might well last a lot longer on a red cell, just as it lasts a lot longer bound to a dendritic cell. And these may then help to transfer it. So you may get a more rapid progression to AIDS if you are unlucky enough to get infected in that way. So what could we conclude? The recapitulation? Well, I think we can say our knowledge of receptors does give us insight into pathogenesis and opportunities to develop novel therapeutics, hopefully microbicides and hopefully vaccines. I would also argue that humoral immunity may have a role in vaccine design, because the whole weight of opinion amongst immunologists trying to make vaccines against HIV is cytotoxic T cells. And Andrew McMichael gave a very nice Ernest Chain lecture about that. Was it last year or the year before? I came along for it, and I'm sure T-cell immunity is important, but humoral immunity and neutralizing antibodies tend to be sort of put on the back burner. And yet, if we think of the successful viral vaccines that are out there, we know that they correlate with neutralizing titers of antibody. It doesn't mean cell-mediated immunity doesn't play a role, but we do know there's an immune correlate with neutralizing antibodies. So I'd like to stay in business on that one. Now, there are some people, of course, who say all this science is a waste of time. We ought to behave more decently. The White House, for one. So I've got nothing against promoting ABC, but simply sticking up a no-sex sign isn't very efficacious. if these were patients on a drug, we'd be saying it's very difficult to adhere to the regime of abstinence. So we do need safe and efficacious microbicides and vaccines if we're going to protect the next generation. These kids are sitting outside the outpost of the MRC labs in the Gambia in Cairo in Guinea-Bissau. I took it several years ago. They've probably grown up by now. So for the Coda, what I haven't mentioned is how we measure neutralization and how we measured receptors. And very often we don't use the live HIV. We use what we call pseudotypes, which we now base on HIV itself, or what we call lentiviral vectors, because no one likes to think of them as HIV, which have been pioneered at UCL by my colleagues Mary Collins and Yasser Takeuchi. So for years we've been using the G protein of VSV on these vectors. You can assemble a completely foreign protein onto HIV and put a reporter gene inside and get a functional entry assay. And we do this all the time in our labs if we want to transfer genes, use a lentiviral library, or mouse leukemia virus. You've got replicating cells, works just as well. We've used that. But we could also say, because they've got these envelopes, we can study neutralization, and that's what we've done with HIV and HTLV. First, we use vesicular stomatitis virus itself, and then after retroviral vectors were developed, we switched to those. So why not use them for real nasties like SARS and influenza? And that's exactly what we've done. So some work of Nigel Temperton, first starting with SARS, was to take the gag pol of HIV, or MLV, but the envelope glycoprotein, the S protein spike of the SARS coronavirus, and put in a reporter gene, in this case green fluorescent protein, and you get this hybrid virus that's only infectious for one cycle. It can get into cells, deliver its gene, which is expressed, and then it's dead. And this is what we call a pseudotype. In this case, I would call it a sheep in wolf's clothing, because this is a very nasty virus, the SARS virus, and you'd think a hybrid with HIV is even worse, but it's safe because it only works for one cycle. So you throw these 3 DNA plasmids into cells, out comes the pseudotype, and you can assay it on cells, provided you've got cells that express a receptor. In this case, it's angiotensin converting enzyme type 2 happens to be the receptor for the SARS coronavirus, discovered very quickly. Well, you can subject these to neutralization, just as we do with HIV in its own coat, and with no antiserum, you get infection, and each cell that's infected will light up if it expresses its green fluorescent protein. Neutralization, complete neutralization, you won't see anything. So it's a neat little assay. Here's what we found with SARS patients. We had 36 patients, but only half of them survived. By 2 weeks, all of those that have survived have neutralizing antibodies, and they maintain their neutralizing antibodies. And we just completed a much bigger study together with Chris Lee in Drowning Through in Oxford in Andrew McMichael's unit, and Gavin Screeton, Imperial's collaborate on this, where we found that in these recovered patients, only about 40% of them show cell-mediated immunity with CD8 responses, but almost all of them have good neutralizing titers, as you would expect for a virus like this. And you can do the same with flu. You can take a highly pathogenic H5N1 flu, put the hemagglutinin in on the retrovirus, and you can look at assays. In this case, we've worked with a lot of people, with Maria Zambon at the HPA, Ian Brown at the VLA, with Chicken Sera, John Wood at NRBSC, who provided the HA plasmid, and Jeremy Farram at the Wellcome Trust unit in Vietnam, who provided most of the patients who'd actually had H5N1 flu. And we get a very good correlation with hemagglutination inhibition, less good with Maria's microneutralization, probably because neuraminidase is also a target for neutralization, but it's still pretty good. And it's more sensitive than either. And it's safe. So you don't have to handle the SARS virus, which has escaped 3 times from labs to infect people and kill people. You don't have to handle H5N1 flu. There's a big debate at the HPA, Mill Hill, and NIBSC. How many Category 4 labs are we going to have? Well, a lot of this work can be done without Category 4 labs, simply by using pseudotypes. And we're thinking of doing the same with rabies, because all the work has to be done in Category 4, the VLA. Now, if VSV works, rabies is going to work. So it's just a new twist, but people haven't thought about it that way before. So that's my story. I'd like to thank the present members of my lab. Bit depleted in numbers here because 2 of them on maternity leave, which doesn't do great for the research assessment exercise, I can tell you. But we're managing. I've had long-term interactions and collaborations with other colleagues. Barry Collins, who's here as my head of department, Yasu Takeuchi. 2 people came to my lab as technicians and stayed for 18 years. Both have moved chairs. So Paul's gone to UMass in Worcester, Massachusetts, and Anya, who's here tonight, has gone to Queen Mary. I miss them both, but I'm very happy for their careers. And I'd like to say, throughout this whole 20 years plus of working on HIV, my work's been supported by the Medical Research Council. I always curse them and argue with them, but I love to bite the hand that feeds me. But I'm really grateful to the MRC, and I'm the 3rd of 5 trained lecturers, I think, who's been mainly funded by MRC. Because there's John Scale and Andrew McMichael, who MRC staff. That's not to knock any other funding agency. I just happen to have benefited from this through competitive grants that have allowed me to do this work. So that's my story. Thank you.
You've been listening to a special lecture vodcast from Imperial College London.